mutant did not recruit galectin 8 at any given point of time. We again found out whether there is this bacteria was actually interacting with galectin 8 as well as NDP52 and LC3, and you can see a nice co-localization of all these three markers. So almost all 90 up to 98% or 100% of the galectin 8 positive bacteria are LC3 positive. That means all those bacteria which were puncturing holes on the endosomal membrane and therefore able to recruit galectin 8 were actually able to recruit LC3. That means it is getting diverted towards the autophagic mechanism. All right. And then in turn that galectin 8 particular bacteria was almost 80% of the galectin 8 positive bacteria was positive for the LAMP1, which is the marker for the lysosome. So that means not only it is getting diverted towards the LC3 compartment, those LC3 compartmentalized bacteria was further getting directed towards LAMP1, which is the lysosome specific marker. So having said that, that means galectin 8 is pretty much central point in terms of present over there, and if we knock down galectin 8 in some way or the other, then there should be an increase in the bacterial count. So that's what we did. We use an siRNA, and indeed what we see is that the LC3 GFP positive bacteria substantially goes down in the galectin 8 knockout cells. So galectin 8 was central in terms of driving the bacteria towards the autophagosomes, and again under this galectin 8 knockdown condition, there is a substantial or significant increase in the intracellular bacterial count of Streptococcus pneumoniae in those HBmac cells. So galectin 8 is the central player which was actually sensing the pneumolysin causing pores, pneumolysin mediated pores on the endosomal membrane, and therefore driving it towards the autophagosome, and those autophagosomes were getting fused with the lysosomes in order to getting it degraded. So that is fine. Up until this point it was very clear. Now whether this particular thing, as you can see, this is a classical path of autophagy, and there is this damaged organelle, there is some bacteria and all that, and this is how it starts. It starts with a phago4, and these are the molecules such as ULK1, FIF200, they come first, and they first starts forming this phago4 isolation membrane, and this finally it closes up and fuses up with the lysosome. That's the total autophagic process. So that means if at all we can target this couple of genes over here, which is the ULK1 and FIF200, they will tell us about this thing, whether this is a canonical autophagy or a non-canonical autophagy, whether there is involvement of this pre-initiation complex. Now to our surprise, when we actually knock down these genes, that is we using siRNA, this is FIF200 and ULK1, we did not see any decrease in the bacterial survival. So, and also moreover, there was no difference in the GFP-LC3 co-localization with the bacteria. Understandably, you would actually expect that under knockdown condition there will be less number of bacteria that will be targeted towards this autophagosomes, but we are not seeing any difference over there. So that particularly tells us whether pneumolysin induced autophagic targeting is independent of autophagy pre-initiation complex. That means whether it is a canonical autophagy or a non-canonical autophagy, and we are working on that aspect particularly to find out whether this bacteria is actually contained within the single membrane structures or double membrane structures, because autophagosomes generally contains of double membrane structures, and we think since it is a non-canonical form of autophagy which is generally given by a particular form of endocytosis called LC3 associated phagocytosis or LAP, we believe that it is because of that, and that's why it is a non-canonical form of autophagy and not classical autophagy. Beyond this line, I would like to show two videos over there which will give you an idea about what is the galectin 8 is doing. So let's look at this. Okay, let's look at this particular thing over here. There is a bacteria which is LC3 GFP galectin 8, and you can see there is a bacteria over here, and slowly the red signal is not visible, but the red signal will slowly go fade over there. After a certain time, the red signal will get fading. So that is one of the population of the bacteria. Unfortunately, the other video will not play up great because of the fact that there is no red signal that is being visible over here, and no, you cannot see that. You will not be able to see that. Let's not. It can be seen over here, but not over here. So okay, but so there are two phenomena which comes over there, which is unusual, very unusual in fact. That one of the phenomenon, the bacteria was wild type bacteria, which was the pneumolysin was targeting it towards the autophagosome, gets degraded into this galectin 8 recruited compartment. So that means it was getting targeted towards autophagy and then finally getting degraded by autophagy fusion with the lysosome, and that's what you can see. And this is the IV versus IC value, that the intensity of the fluorescence in the phagosome ratioed by intensity of the fluorescence in the cytosol. And as you can see, if at all the intensity of the fluorescence in the phagosome by the intensity of the fluorescence in the cytosol becomes 1, that means it is already gone. There is nothing over there. So you can see over here that the galactin signal is still over there, but the bacterial signal, which is YFP, tag RFP, which slowly comes to the 1. So that means the bacteria gets degraded in the galactin 8 recruited compartment. Contrary to this, look at this graph over here. The GFP signal goes very quickly away. But I unfortunately cannot see, but from this graph you can realize that GFP signal comes to 1 very quickly, but the bacterial signal or the tag RFP signal remains over there. So that means there are some bacterial population which is able to escape out of this endosomal structure, which is phenomenal to us, we were not even expecting this. We thought like, okay, this bacteria would go inside an endosome. There will be pneumolysin mediated pore formation that will recruit galactin 8, which will in turn target it towards the autophagy and finally get degraded. Story ends over there. But the story took a new turn over there, in the sense we were watching that some population of the bacteria were able to come out of the endosomes. Now, what was happening to those particular population of the Streptococcus pneumoniae which were able to come outside of this? We actually quantified it and actually see that approximately 63% of the wild type bacteria gets degraded inside the vacuole, and possibly by autophagic mediated mechanism. But approximately 37% of the bacteria, these are all quantification of the live imaging data, that we can say that 37% of the bacteria come into the cytosol. Now, what might be the mechanism of degradation of a bacteria which is lying free in the cytosol? So this is what is happening possibly. Wild type bacteria getting in because of virtue of this production of pneumolysin, it damages the endosomal structure that recruits these blue molecules which are galactin 8, and those galactin 8 mediated molecules either fuses with the lysosome, that is called autolysosome, and that gets degraded. But 37% are actually following this mechanism where they further degrade the L double membrane structure and then finally comes over here in the cytosol. Now very recently there are people have proposed that once the bacteria is free in the cytosol, there is ubiquitination machinery happens on the bacteria. So ubiquitination then further degrades the bacteria by recruiting again autophagy. So another further backup mechanism for the cell. So what I am trying to emphasize over here, that not one, not two, cell has multiple backup mechanism in order to take care of the invaded pathogen. Once it was in the cytosol, once it is in the endosome, it can get degraded by fusing with the lysosome. Once it is still can break the endosome, then it can get fused with the galactin 8 and finally get targeted towards the autophagosome. But still, if at all it can come on the cytosol, cell has this machinery over here which is ubiquitination followed by autophagic capture. So we were thinking, okay, what was happening to the wild type bacteria? You see the wild type bacteria gets ubiquitinated over there, but not the mutant bacteria. Mutant bacteria does not even recruit any ubiquitin signal. So you block ubiquitination by using this PYR41, an inhibitor of the E1 ligase, and you see higher bacterial survival. So ubiquitination in fact was responsible for killing of the bacteria, and these confocal micrographs exactly show that also. And this is very curious. So we see this, there are three different populations. One population was only galactin 8 positive, there is no ubiquitin. So that means that particular population of the bacteria was the 63% bacteria, there was no ubiquitin recruitment over there. Then there was a second population of the bacteria which was both galactin 8 positive as well as ubiquitin positive. That means probably a little bit farther damage on the endosome, and that ubiquitin signal was getting recruited on there. And then the third population, which undoubtedly proves that the bacteria is present on the cytosol, is galactin 8 negative but ubiquitin positive. So that means the same bacteria, what I am trying to project over here is that heterogeneity of the bacterial population. What we have infected the cells with not three different kinds of bacteria. We have infected the cells with a only single kind of wild type bacteria, but once it goes inside the cells, it starts behaving differently. It starts behaving differently means it starts behaving differently because of the fact that it can express different quantities of pneumolysin. So that means there is a plethora of different population that might exist of this only one type of bacteria inside the cell just because of the expression of only one virulence factor. This is pneumolysin. So we have quantified this, and you can see with increasing time point, you have more only ubiquitin positive bacteria present in the cytosol and not much galactin-8 positive bacteria. And there are some bacteria which are both positive, and there are some bacteria which are only galactin-8 positive but ubiquitin negative. So there are three different populations, and this PLY mediated pore formation was at the central point of all this phenomenon, because PLY, if at all you do not have, neither it recruits galactin-8, neither it recruits ubiquitin. So PLY is central, and possibly this is what is happening. There is gradual pore formation what is happening over there. Slowly one pore forms, and that's what we believe, that small pore actually recruits galactin-8, and larger damage, that means larger pore formation, actually recruit ubiquitin. And from this slide we can say that SPN displays, SPN means Streptococcus pneumoniae, displays heterogeneity in PLY expression. So that means the bacteria that has gone inside, following after going inside, it can have heterogeneity. And now you can see from here that the ubiquitin positive bacteria which were present only in the cytosol, they are almost 80% of the ubiquitin positive bacteria are present in the cytosol, and they show co-localization with the LC3. That means they are again getting targeted towards the lysosome by an autophagic dependent process. So in order to tackle this heterogeneous expression of pneumolysin in the wild type bacteria, what we did is that we fused GFP transcriptionally with this PLY, and then this where is that called a FACS analysis. And you can very clearly see there are some majority of the bacteria actually are GFP negative, so they are not even producing pneumolysin per se, but there are some bacteria, approximately 45%, which are producing GFP, out of which some are producing exceptionally high quantities of GFP, which is over here, and some are producing intermediate, and majority of them are actually very low GFP producer, that means very low pneumolysin producers. So we have a population where we have some high bacteria produce high expressing pneumolysin, some low expressing pneumolysin, and some intermediate expressing pneumolysin. So how to tackle that? So very recently there was a paper in 2014 where people have done some mutations at the 5' UTR of the pneumolysin gene, and by which they can actually regulate, they are basically making the mRNA secondary structure less stable or more stable in order to increase or decrease pneumolysin expression. We adopted the similar mechanism, and we actually put pneumolysin high and low in the genome of Streptococcus pneumoniae, and this is where you can see that low strain and the high strain, there is a considerable difference in the pneumolysin expression. And to our surprise, what you see over here is phenomenal, is once we actually been able to lower the pneumolysin expression in the bacteria, that is actually able to survive much higher in the brain endothelial cells compared to high pneumolysin producing bacteria. So that means our objective was that high pneumolysin producing bacteria was able to escape into the cytosol, but once it escapes into the cytosol, it can get captured by the cellular ubiquitination machinery followed by autophagy, and that's how it can get killed. And actually you can see that high producing pneumolysin bacteria are more ubiquitin positive compared to low pneumolysin expressing bacteria, which is very less ubiquitin positive. That means the cytosolic bacterial population is substantially increased in the high pneumolysin expressing case compared to the low pneumolysin case. So we can say fairly that the lower PLY expression promotes higher SPN intercellular survival, higher PLY expression promotes higher vacuolar escape and further cytosolic degradation. With this, I'll come to this. We have actually designated this for the following endocytosis. Bacteria gets in an endosome, and if there is intermediate production of pneumolysin, there is damage to this, then there is galactin-8 recruitment. And if there is further damage, accumulation of pneumolysin over here, either the bacteria can get ubiquitinated or the damaged endosome can get ubiquitinated. Further production of pneumolysin can actually drive it outside of the endosome. So this is the free cytosolic bacteria which can get degraded either by autophagy, that's what, or by proteasome mediated machinery. On the other hand, this particular population, which is GFP galactin-8 positive, ubiquitin positive, that can actually go to this non-canonical autophagy. Mark my words, because this is a single membrane, that's why I call it non-canonical autophagy. So this is galactin-8 positive, ubiquitin positive, LC3 positive. And we have very recently found out that if there is an extremely small proportion of bacteria that produces, extremely small amount of pneumolysin producing bacteria, that actually creates maybe ion imbalance over there, and that osmotic imbalance in turn recruits LC3. So this particular population is gal-8 negative, ubiquitin negative, as but LC3 positive, and these are probably the longest surviving population inside the brain endothelial cells, and they actually are able to transcytose from the blood side to the brain side. So with this, I will end over here, and I would like to thank all the students who has contributed to the lab, and particularly Manali Surve, whose work I have presented over here, and she is present among the audience, and all my collaborators in IIT Bombay, as well as Professor Quaix Kim, who is present in Johns Hopkins, who has given us this brain endothelial cells. And I thank you all for your kind attention, and I would like to take any questions if you have. Victor. So this is something which we are doing. It's a very basic biological studies. I don't see it going into any sort of any commercial applications of this for this. But yeah, there are several aspects from other sides of on the similar angles that we are working on. It can possibly show that, you know, if at all we can induce autophagy inside host, that can tackle the bacteria, and that's a lot of labs across the globe. They are trying to induce autophagy, particularly with respect to TB infections. That can take care of the intracellular Mycobacterium tuberculosis, which very safely resides inside the pulmonary macrophages out there. Can we trigger autophagy in those cell populations in order to tackle the intracellular bacterial population kill off?
Because the biggest problems are Mycobacterium tuberculosis, pneumococcus, and hospital-induced Pseudomonas and Klebsiella.
Correct.
I mean, these are the major issues with
so this is a relatively new angle which has come into the forefront in the last 10 odd years or so, where cellular autophagic machinery has been described to tackle these problems. And autophagy is generally regarded as a bulk process, which it can take care of, you know, the damaged organelles, damaged protein, misfolded proteins and all that. But nowadays, very recently in the last 2 or 3 years, the autophagy has been increasingly regarded. Can we make it selective? There is something which is called selective autophagy, is it present or not? So there are proteins which are degraded, and that can be taken care in terms of Huntington disease and all that. There is something which is called selective autophagy. Can we actually employ similar sort of mechanisms over here in the bacterial diseases? That's under exploration, of course.
Just one more question.
Sure.
Since there is a major problem of most of the CNS drugs not crossing the blood-brain barrier.
Correct.
Can you use these phages as a carrier to carry other drugs into the brain?
One of the, what was your question? Can we?
No, the problem mainly with most of the CNS drugs, including for Parkinson, Alzheimer, everything, is the resistance to cross the blood-brain barrier.
Agreed.
So if you have got a solution to that problem through this mechanism,
so
can you use them as carriers?
Maybe not
to take other drugs into the brain system.
Maybe not exactly as a carrier, but what you can think of doing is that, can we learn something from these bugs, the smart way that they adopt, and can we actually learn from that particular tactics and employ that in the drug delivery context to take the drug from the blood to the brain? Can we learn something like that? We are trying to, let's see.
So Anirban, great talk. First I want to comment that I think you're illustrating what I think kind of the second wave of innate immunity is really revealing, that where the first discoveries such as toll receptors were focused on the recognition of molecules or patterns of bacteria, and that was the central aspect of innate immunity, is, I think, a little too simplistic, because all the good bacteria and non-pathogens also have LPS and flagella, and you don't want to respond to them.
Agreed.
And what you're showing, and I think other people have discovered for different aspects of pore-forming toxins or translation-inhibiting toxins or broad-spectrum proteases, is that there are cell-intrinsic innate immune defenses that are triggered in response to the recognition of damage to the host mediated by the microbial toxin.
Correct.
And that triggers an innate immune response, which in the case is clearing this. My question is that, you know, galectin in this model is recruited to the damaged membrane.
Correct.
But for all these pathogens that invade the blood-brain barrier, they have polysaccharide capsules, and pneumococcus has many. Is this in the D39 background or a different background?
It's in a D39 background. So what's generally accepted over there, if at all you have capsule, then this bacteria does not go in. And it's been shown by Elin Tomanen's group that this autolysin is there. The moment the bacteria attaches on the eukaryotic cell, this autolysin comes up and degrades, chops up this capsule. So it needs to get devoid of the capsule once it wants to go in.
Right.
So that's an antiphagocytic factor, and as long as it is present, it will not go in. So the autolysin comes at and rescues it in terms of like just by degrading the capsule.
Right.
Now it can go in.
And even the type 2 capsule, I think, is mainly
So
rhamnose and other things, probably doesn't have galactose. But I'm wondering if galectin, in addition in these models with the intracellular pathogen, can also be recruited to the pathogen.
Correct.
Because I think another pathogen that we've collaborated on in addition to the pneumococcus is group B streptococcus, which is much more efficient actually, like in the cellular assays in terms of percentage of bacteria doing this. And they have a capsule in which there's a terminal sialic acid that always masks an underlying galactose, which would be recognized by galectins.
Correct.
So it might be really interesting to see, I don't know if group B strep, I don't think it sheds its capsule completely to invade the organism, but one of the reasons is that maybe the unencapsulated one is targeted directly by the galectins and goes to the pathway you've described, and the encapsulated one, by putting a sialic acid over the galectin, avoids that and is more virulent, and that could be kind of tested in exactly the way that you're doing this model.
Fantastic.
For differential targeting using the asialo mutant versus the wild type mutant. We have a couple of those types of strains for you to study.
Absolutely. Why not? Yeah, I would love to study them. Yes, of course. I guess it's fine.
Thank you very much, sir. I request Dr. Sanjay Pal, Assistant Professor, Amrita School of Biotechnology, to present the memento to Dr. Banerjee. Thank you, Dr. Banerjee. Thank you, Dr. Pal. We now break off for lunch. Join us post-lunch for the much-awaited town hall session on superbugs versus superdrugs, the looming global drug resistance crisis. It shall start at 2 p.m., so please be seated by 1:45. Students, please note, don't forget to put in your chits in the box outside. Thank you.